Hey guys, welcome back to Pharma Palette. So today we are going to discuss about the life cycle of RBC in detail, right from its production till its destruction. So we will also see the complete life cycle with the help of a clear diagram. So it becomes very easy to remember for exams. And at the end of the class, we will also discuss the functions of RBC and their role in immunity. It is very repeatedly asked in your theory exams. As always, I will write the same points that I explained so you can directly use these notes for your exams. So let's get started. So before knowing its life cycle and its phases, first we have to know some basic points about it. First thing is the life cycle of RBC. In this, the lifespan of one RBC will be 120 days and it is mainly originated from the red bone marrow, and the process, it is also called as erythropoiesis. It is simply the process of RBC development from the stem cells. So stem cells are simply the precursor cells or the starting cells from which the RBC is developed. So it generally takes 7 days to complete this process. Now let's try to know the different phases in it. So the life cycle of RBC has three main phases. First thing is production, maturation and destruction. Now let's see each phase in detail. So before entering into each phase in detail, let me just show you one flowchart. I already said that this process mainly starts from the stem cell. So that stem cell name is hematopoietic stem cell. We have discussed about it in the previous videos. If you want, you can go check it out there. So from this hematopoietic stem cell, it will be converting into common myeloid progenitor, and from there it gets converted into megakaryocyte erythroid progenitor. From that the final erythrocytes or the RBCs will be formed. Now let's see each phase in detail. So coming to the first phase, which is the production. So in this production phase, generally the erythropoiesis will be primarily occurring in the bone marrow. Second thing is it will be starting with the hematopoietic stem cell. From there, it gives rise to the multipotent progenitor. Then it gets converted into erythroid committed precursors where it matures RBC. Then it gets converted into erythropoietin. And this erythropoietin is the primary regulator of erythropoiesis, which will be leading to the proliferation of RBC. Once the RBC gets proliferated, it will be preventing it from the erythroblast apoptosis. So that is all about the first phase. Coming to the second phase, which is maturation. In the maturation phase, here it will be undergoing certain morphological changes where it produces the specialized cells which are highly functional. Then this process mainly includes the expulsion of nucleus and loss of cell organelles like rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, ribosomes, everything will be getting lost. Finally, it will be leading to the formation of mature RBC. Once the RBC gets matured, it will be having a shape of biconcave. So the last phase in this life cycle of RBC is the destruction phase. So already we have seen that in the maturation phase, the RBC got matured. Now that matured RBC will be staying in our body for 120 days. Once the lifespan gets end, it will be removed from the circulation and it is destroyed by the macrophages in the spleen and liver. Now let's try to know this complete process with the help of a simple diagram. Consider three parts here. So this one is macrophage, this part is liver, and this part is the bone marrow. So this is the complete diagram of erythropoiesis. Let me just explain it to you very clearly. First thing is, see the steps and the numbering, then it will be easy. Blue colored part is macrophage, that is present in spleen and liver, and this part is called as liver. and this is the bone marrow part, and this is the small intestine part. So we have seen the different organs now. Let me just start with the first step. So once the 120 days lifespan of the RBC is completed, that RBC will be getting destructed. Once it gets completely destructed with the process of phagocytosis, it will be releasing two things. First thing is the heme part, second one is the globulin part. So this globulin part will be converted into amino acids. That amino acids will be changed, they will be used for the protein synthesis. So that is the globulin part. Now coming to the heme part. Heme part generally is having Fe3 plus. So with the help of transferrin enzyme, it will be converting this Fe3 plus to ferritin in the liver. And also in the liver, again it will be changed into Fe3 plus with the same enzyme transferrin and gets transferred into the bone marrow. And in the bone marrow, it will be having globulin, along with that vitamin B12 and erythropoietin. By using all these elements, it will be undergoing the process of erythropoiesis. Once the erythropoiesis occurs, it will be forming the RBC, and that RBC will be having this 120 days life cycle. And here this is the production of RBC, and there is some other part happening here. Here we said that it is divided into heme and globin. This heme part, it will be also converting into biliverdin. This biliverdin will be changed into bilirubin and it is transferred into the liver. Once it gets transferred into the liver, it will be sending that to the small intestine. In the small intestine, this bilirubin, with the help of some other bacteria, it will be changed into urobilinogen. This urobilinogen will be entering into the urobilin, and this urobilinogen can also convert into stercobilin, and from this stercobilin it will be releasing the feces. So the stercobilin is responsible for the color of feces. Similarly, after this urobilin is formed, it will be excreted in the form of urine. So that is the simple process of erythropoiesis. So coming back to the some other functions of RBC, first thing is it is useful for transporting of different respiratory gases. It can be oxygen or carbon dioxide throughout the body. Second thing is this hemoglobin will be binding to the oxygen and forms oxyhemoglobin. And this oxyhemoglobin will be responsible for transporting 97% of oxygen throughout the body. Similarly, if the hemoglobin gets binded to the carbon dioxide, it will be giving carboxyhemoglobin, and this will be also transferring approximately 30% of carbon dioxide. And this is useful for contributing and maintaining the acid base balance in the body, and this is also useful for the blood group determination. Coming back to the last part, which is the role of RBC in immunity. And this RBC is useful for the binding and scavenging process. It is useful for the phagocytosis. This is also useful for producing the reactive oxygen species. So this is also used in the process of immune recognition. So that is all about the life cycle of RBC along with its diagram, their functions and their important role in immunity. So these points are extremely important for your exams. So make sure to revise the notes that I wrote along with the explanation. In the next video, we will discuss another topic. So if you found this video helpful, don't forget to like, share and subscribe to Pharma Palette. See you all in the next video and thanks for watching.